welcome back for the last video. Recall the previous problem about OptiGrow. The makers want to ensure that their product will work under a variety of watering conditions. Maybe we should include the amount of water as part of our experiment. So remember, there's always, there could always be more than one variable. And water amount is another variable, right? Which we call a factor. Can we study a second factor at the same time and still learn as much about the fertilizer? So if we do include water, we now have two factors now. We've got fertilizer at 3 levels. So fertilizer is our one factor, and then we'll call the water irrigation. And that could be at 2 levels. Either they're getting water or they're not getting water, right? We combine them in all possible ways to yield 6 treatments, 3 times 2. And here you can see the treatments listed out. So there would be 6 groups, right? 6 treatment groups. You have a group that has no water and no fertilizer, no water and half the fertilizer, no water and full fertilizer. So none of these groups have any water, but they're getting the treatments. These groups get water, but they're also getting the same 3 treatments. If we allocate the original 24 plants, the experiment now assigns 4 plants to each of those 6 treatments at random. And we call this experiment a completely randomized two-factor experiment. Because any plant could end up assigned at random to any of the 6 treatments. And we have 2 factors. It's often important to include several factors in the same experiment in order to see what happens when the factor levels are applied in different combinations. So here, when you take the extra factor of irrigation, you can see here that we added no water to those 3 treatment groups and then water to those 3 treatment groups. Please beware though, when the levels of one factor are associated with the levels of another factor, we say that these 2 factors are confounded with another. So here's an example. After many dogs and cats suffered health problems caused by contaminated foods, we're trying to find out whether a newly formulated pet food is safe. Our experiment will feed some animals the new food and the others a food known to be safe, and a veterinarian will check the response. Why would it be a bad design to feed the test food to some dogs and the safe food to some cats? So this would simply create confounding. We wouldn't be able to tell whether any differences in the animal's health were attributable to the food they had eaten, because the dogs were getting the test food and the cats were getting the safe food. And, the differences in how the 2 species responded would be confounded in that way. And then I'm also going to add in here were attributable to the food they had eaten. Or to differences in how the 2 species responded. There's a 2-factor example that you can read through. That's a good example that also talks about confounding. Now, confounding might sound a lot like lurking variables that we talked about in chapter 6. Confounding may remind you of this because, like lurking variables, they interfere with our ability to identify what's causing an observed outcome. However, there is a difference in how the interference comes about. A lurking variable is usually thought of as a variable associated with both x and y that makes it appear that x may be causing y. So remember, we had that example when ice cream sales increase, drownings increase. But we talked about like ice cream doesn't cause drownings. It's really a temperature could be a lurking variable. On the other hand, confounding can arise when experiments, when some other variable associated with a factor has an effect on the response variable. Okay, remember the response variable is what we're trying to measure. A confounding variable is associated in a non-causal way with a factor and affects the response variable. Because of the confounding, we find that we can't tell whether any effect we see was caused by our factor or by the confounding variable, or even by both working together. One of the roles of blinding an experiment is to combat confounding. And then there's a legendary example of confounding, which you can read through to get an idea. And that's about it for this chapter. If you have any questions, please reach out to me.